Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last few weeks, we have talked about a lot of applications of next generation sequencing, and these revolve around single nucleotide polymorphism analysis or copy number variation analysis, and then we talked about transcriptome analysis, right. So, where we analyzed RNA-seq data to actually compare gene expression levels across different samples from different conditions or different tissue types, etc. So, all these analyses rely on the reference genome. Okay. So, we talked about mapping against the reference genome in case of single nucleotide polymorphism analysis, or in case of RNA-seq data analysis, we are also mapping against reference genome, right. So, that required that reference genome. Now, the question is, how does this reference genome come about, right? Where do you get this reference genome from? Okay. So, this is what we are going to talk about in this week, right. So, the topic would be genome assembly. So, how do you take next generation sequencing data and actually build those reference genomes? Okay. So, that is what we will start with today. So, the concepts that we will cover today revolve around de novo genome assembly problem. So, we will first talk about the genome assembly problem, right. So, how is it different from the mapping problem, right. So, we talked about mapping, whether it is RNA-seq or whether it is targeted resequencing, right. So, there was this mapping, and we talked about algorithms that actually help in the mapping process. So, similarly, we have this genome assembly problem, right, and especially the de novo genome assembly, right. So, we are building this genome from scratch, right. So, we do not have any comparison or anything. This is a very difficult problem. So, we will first elaborate this problem, right, and so that you understand, right, how difficult this problem actually is. Okay. And then we will talk about some ways to address this assembly problem, right. So, how do you go about assembling genomes from the reads that we get from the next generation sequencers. So, we will talk about one such approach today. It is called shortest common superstring approach, or SCS approach, and then in subsequent classes we will talk about other approaches. Okay. So, the keywords that will come across today are overlaps and directed graph. So, let us begin, right. So, with the flow chart that we started with. Okay. So, here we are, we have the read data, we do the data quality control. So, this step is common across all the analysis. Then we talked about the left part mostly, right. So, we talked about this read mapping and then genome analysis and transcriptome analysis. We have not talked about epigenomic studies yet, which we will cover in the last week. So, the other part that is remaining is the read assembly part, right. So, this is this is the part which we have not discussed yet, okay, and this is what we are going to talk about. So, the part in green, those are complete, right. So, we have discussed those in this course. The part that is that we are going to discuss today is the read assembly part, and then we will follow this up for the subsequent classes. Okay. So, let us now understand the assembly problem, right, and why it is a challenging problem. So, here is the simple schematic which is showing DNA, right, which is first fragmented into small fragments, and then they are processed according to this adapter ligation, amplification, etc., depending on the platform. We have discussed those numerous times. Okay. So, I am not going to go into that. So, once we get these fragments, we process them through NGS, and what we get are the sequenced reads. Okay. So, these reads are shown in blue color here. Okay. Now, once we have these reads, the assembly means from these reads we would have to actually generate the sequence of this DNA that we started with. Okay. We have no idea about the sequence of the DNA that we started with, and we want to generate that sequence. Okay. The order of these sequences, right. So, each read probably will have some unique sequence, right, and how they occurred in the genome, right. So, we have to kind of order them, right, in and then find out the full genome sequence. Okay. So, that is the problem, right. This is the assembly problem. We have to get the reference genome sequence. Of course, I am showing this as one single stretch of DNA, it is not, right. If you talking about eukaryotic or mammalian genomes, right, to have multiple chromosomes. So, the idea is to get those chromosome sequences from this read data. So, this is the problem. Okay. Now, let us understand the challenges, right. So, with a very simple analogy which we all are familiar with. So, we must have played these puzzles, right. So, these pieces, right, that you have to put together to get a picture, right, to generate a nice picture. Okay. So, these are the pieces of the puzzle. So, you can imagine these are like reads, okay, which you would have to put together. Right. We have fragments of this genome which you have to put together to generate the full genome sequence. Now, this when you put them together, this will look something like this. Okay. But then one thing you should remember, right, when you do these puzzles, you have a reference picture somewhere, right. So, you look at the picture and say, okay, this seems to fit with this seems to fit with that, right. So, then you can put this together. When you are doing the assembly, you do not have that reference picture, right. So, you have to put them somehow put them together that they makes sense, right. So, you have rough idea what you are going to see, but then you do not have any reference image, right. So, you can now understand how difficult this problem is, right. So, if I give you, let us say, this even 1000 pieces of a puzzle and I say, okay, assemble them so that they make some sense, right, they make some nice picture, but I am not going to give you any reference picture or reference image. Okay. So, that is what the assembly problem is. We have no idea about the reference genome sequence. Okay. We have some sort of kind of maybe number of chromosomes, etc. we can guess, right, we can know, right, by other methods, but apart from that we will probably have no idea about the sequence of the genome. Okay. So, without looking any picture or any reference, we have to put them together that they make some sense. Okay. So, this is what the assembly problem is, and that is why it is incredibly challenging. Okay. So, let us now work with very small DNA sequences and then try to understand this problem and see how we can solve this, right. So, once we go into this understanding, right, once we understand what this problem is about, we can think about solutions, right. So, how we can approach this problem and try to solve this. Okay. So, let us take this example where we have this reference DNA, it can be any DNA, right. So, I have just taken one sequence and we take this sample, right. So, when we take this sample, we have this DNA in multiple copies from multiple cells, right. So, let us say you are working with a population of cells, right, and you extract genomic DNA and then you sequence, right. So, in a sense you have multiple copies of the same genome sequence, okay, maybe with some minor differences here and there, but most of them are identical. Okay. So, you generate these multiple copies, here I am showing only 4 copies, right. So, you can imagine this can be millions depending on number of cells you are working with, and then we fragment. Okay. So, the fragmentation process will generate random fragments, right. So, it will fragment at random places mostly unless you are using restriction enzymes for digestion. So, any other such as physical fragmentation, right. So, those will generate random fragments. Okay. So, I have shown these fragments by these spaces in between, right. So, we have these for this copy, let us say we have this fragment, one fragment here, then the second fragment, then the third fragment, and it is very unlikely that you will get fragmentation at the same place for all DNA copies. So, that is why you see different types of fragments. So, what we will do is we will take these fragments, right, we process them through this adapter ligation step, amplification, and maybe there is some sort of size selection where we might remove very short or very long fragments depending again on the platform that we are using, right. So, there will be an appropriate size and then we sequence these fragments. Okay. So, now what I will say, let us say we generate these sequence reads and we need to generate this input DNA sequence. Okay. Now, if I arrange these sequence reads like this in this order, right, we can see, okay, this read kind of overlaps with this one, and if I arrange these reads in order, it is easy to generate this assembly, right. So, it is easy to generate this input DNA sequence, right. So, if you look at this and say, okay, yeah, that is it. So, this would be the reference sequence or the input DNA sequence that we started with. Now, the problem is, of course, the reads will not be ordered like this, right, when you get from the sequence, okay, and they will look something like this. Okay. Now, when you have this kind of order, right, so this kind of random fragments, right, and random sequences, how do you actually generate that input DNA? Okay. And even with this small example, right, where we have only a handful of reads, right, you can see this, this becomes quite a challenging problem, right. It is not so trivial. Again, of course, there are ways you can now start to think about, right, how we can solve this issue, right. So, you can imagine, right, with millions and billions of reads, this will become very, very difficult as well as time consuming. Okay. So, let us think about some approaches, right. So, the question is how can we do the assembly, right. Let us start with a very simple example that I showed you, and then think about, right, how we can actually extrapolate that to situations where you have millions or billions of reads. Okay, so, there are three types of assembly algorithms that we will discuss, right. So, these algorithms have been designed to do this task precisely, right. So, to generate this reference sequence from the read data. Okay. So, these algorithms are employed for this purpose. So, the first one we will discuss is called shortest common superstring approach or SCS approach. The second one is the overlap layout consensus or WLC approach. The third one is deep brain graph approach or deep brain graph assembly. Okay. So, we will discuss each of these approaches one by one. Okay. So, we will start with the shortest common superstring approach. Okay. So, this is derived from this idea in computer science, right. So, you have this superstring. Okay. If you are given some strings, you can have superstrings. Okay. So, what are superstrings or what is a superstring? So, it is a string, right, that contains all given strings as substrings. Okay. So, we will take an example and that will be easy to understand. Okay. So, let us take this example here, very simple one. We have 4 strings, let us say we have given 4 strings. Okay. So, rain, day, cloud and sunny. Okay. Now, for superstring, right, this will contain all these strings, okay, and these will be then called substrings of the superstrings. Okay. Now, you can generate superstring very easily if you just concatenate, right, if you just put one after another, right. So, that is an easy process, right. So, you can simply put them together and you can see, right, you can find this string rain in this superstring. So, this is the superstring here, you can see you can find rain here, you can see you can find day here, right, you can find cloud here, you can find Sunday here, right. So, this is an easy way to generate this superstring. Okay. Now, what is tricky is you can probably notice is called shortest common superstring or the shortest superstring. Okay. So, shortest superstring is the superstring, right, is the shortest one that contains all given strings as substrings. Okay. So, out of all superstrings that you can think of, this will be the shortest one. Okay. So, this becomes tricky now, right. What is the shortest one? Okay. And that is likely to be a difficult problem. Now, let us go back to the same example, okay, where we have these strings rain, day, cloud, Sunday, right. This is the superstring, right, that we got. The shortest common superstring you can see for this simple problem, right, simple example, you can see this rain, cloud, Sunday. So, where is the day? Because day is already contained in Sunday, right. So, we do not need to again add day here, right. So, we can have these only Sunday and that will already include day. Okay. So, this is the shortest common superstring. So, you can now imagine, right, with given different combination of words, you need to find these superstrings shortest ones, right, and this might not be very trivial as you expand this number of strings, right. So, as you increase the number of strings, this might be a difficult problem, right. So, you can think of in terms of reads now. Okay. So, these strings that are given, these are the reads. Okay. In our case, these are the read data. Okay. Now, from this read data, if we can find out the shortest common superstring, we can perhaps get close to the reference sequence, right. So, that is the idea, reference sequence will contain all the reads that are generated, right. So, this is the idea that with which we actually take the shortest common superstring approach. Okay. So, let us now go into the shortest common superstring with reads. Okay. So, let us again take very simple example with 4 reads each of length 4, right, 4 bases. So, you have ACGA, T, CCGC and CGAT. Okay. So, the superstring, if we just define, we can concatenate, we can just put one after another and that will be the superstring. What will be the shortest common superstring? Okay. It's a bit tricky now, you can probably figure out. One thing is that this part is common between these two reads, okay, SCG and CGAT, and perhaps you can have this SCGAT that will actually contain both these reads, okay. And for the other two reads you have to have these separate fragments, right, because they are unique. Now, as we increase the number of reads, these kind of situations will be more, right. Here we have just one situation where we see this sharing of sequences between these two reads, but as we increase, as we go into millions and billions, we will start seeing a lot of these sharings or overlaps as we call them. Okay. So, this will make the problem more challenging. Okay. Now, how do you actually find these overlaps? Okay. So, where do you start? Okay. So, as you can probably guess now is that we start with the overlap part, right. So, if we have reads coming from same region of the genome, they are likely to show sequence overlap, right. So, that is the idea. Okay. So, if you take a fragment from this genomic region and let us say you have two reads that are coming from this same region, right, they are likely to show overlap, and here in this example you can see this overlap here, AGCAG, this part is overlapping between these two. Okay. So, with this idea we can now continue, right. So, what we do is we represent reads in a directed graph that actually tells us about the overlap. Okay. So, which we also call as overlap graph, right, because this directed graph tells us about overlaps between the reads that we have got from the sequencer. Okay. And as part of the graph we have nodes, right, or vertices, right, as we have discussed very briefly, right, the graph what you have in a graph. So, we have node and edges, right. So, each node is a read, right. So, a read is represented as a node in the graph, and then two nodes or reads will be connected by an edge if there is overlap in sequence between these two. Okay. Now, this is a directed graph, which means you also have directions. Okay. So, you have arrows from one node to another. So, how do you decide these arrows or these directions? Okay. Whether it should go from node 1 to node 2, right, assuming this node 1 and node 2 they are showing overlap. So, would the arrow go from node 1 to node 2 or node 2 to node 1? Okay. So, that is something we also need to decide. Okay. How do you decide that, right? How do you decide this direction of this edge? So, this is decided by the following, right. So, if you are seeing overlap between read 1 and read 2 and suffix of read 1 is showing overlap with prefix of read 2, then we will add the direction of edge from read 1 to read 2. Okay. So, read 1 would be the starting node and read 2 would be the end node, okay, for the arrow. So, the direction would be from 1 to 2. Okay. So, let us take this example, right. So, where we have seen this overlap between these two reads, okay, and for the first one this part is the suffix, right. This is towards the end, right. So, again remember these definitions of suffixes, right. So, we talked about the suffix tree, etc. So, here this is the suffix of the first read here is showing overlap with the prefix of this second sequence here. Okay. So, that means this will be the node from which the arrow will start, and this will be the node from which where the arrow will end. Okay. So, that is why you see now the direction of this arrow here. Okay. So, what you need to do is we need to do this for all pairs of reads that are present in the data. Okay. So, we have over a set of reads now, okay, and this is the input DNA again, and let us say we have this very nice pattern of reads. We have got all the reads that are possible of certain length. So, here we are taking length 6 spaces, okay, just for our example, and we have got this nice overlap. We know these are the overlaps, etc. We will talk about how we actually find overlaps between reads in reality, right. What are the methods? I will just briefly mention them probably in the next class, and then you can utilize those methods to actually find the overlaps between the reads. Okay. So, what we do is we start with this pairwise comparison between reads for overlap. Okay. So, what you do is compare this read 1 with read 2, okay. Then you go to read 1 and 3, right, and you go on, right, you continue this for all the pairs that you have in the data. So, as you can imagine, this will take time, okay. As you have millions or billions of reads, all pairwise comparisons, that will take a long time. Okay. So, of course, these overlap graphs are time consuming. Okay. So, you have to build them in case you have millions of billions of reads, then it will take time. Okay, so again we are, when we check for overlap, we also check for this prefix and suffix part, right. So, which read where the overlap is, whether it is in the suffix part or the prefix part, right, because this will decide the direction of the edge in the directed graph. Okay. In addition, we also have something called cutoff length for overlap, right. So, we do not say just if you have just one base overlap, right, we do not consider that as an overlap or two base overlap. We need certain amount of overlap, right, just to avoid any spurious overlaps, right. So, or overlaps just by chance. One base overlap will happen just by chance, or few base overlaps will happen by chance. So, you need to decide on a cutoff length. So, as you will see in these examples, as we go on, we will choose these cutoff lengths, okay, and then we will draw the directed graph or generate the directed graph with all the directions or the arrows, etc. Okay, so, if we continue this process, we will see we end up with something like this. Okay, so, here I have taken this cutoff length for overlap as 3. So, we need at least 3 base overlap between two reads, and then again we know the suffix and prefix part for each read, and depending on that we add these arrows. Okay, so, you can derive this yourself, and I encourage you to do so with a pen and paper. It is pretty simple. If you just sit around and do this pairwise comparisons, right. For example, these two, right, you can see this will be the direction of the arrow, and then you see the overlap is about 5 bases. Okay. So, you can do this for all of them, and then you can simply add these arrows for these overlaps. Okay. And also we have this, you can see two different colors, they are not actually different, they are just shown for visualization purposes. Okay. So, you can see them better, these nodes. Okay. Some of these arrows are going through the nodes. Okay. So, it looks quite complex, right, for a small number of reads it is already quite complex. So, as you can imagine, if we work on a large number of reads, this will become really complex and it will be a huge graph. Okay. Now, let us move on with this example. What do we do after we have built this directed graph? So, we also add the edge weights to the graphs. Okay. It is not just this directed graph, we also add edge weights to the graph. Okay. So, what are these edge weights? These are actually given as a length of overlap. Okay. So, we can add these numbers now to this graph, which is showing how many bases are overlapping between these two reads. Okay. So, the nodes you remember, these are the reads. So, this is lead 1, etc. right. So, you have all the reads represented as nodes, and you have the edges, and then you have the edge weights, okay, showing the number of bases that are overlapping between these two reads that are connected by an arrow. Okay. So, what is the purpose, right, and what are we going to do with this directed graph? Okay. So, again you can derive this quite easily, right. So, you can at home you can try to derive this from the information, right, of the read sequences that we have just seen, and then you can derive this yourself. Okay. With the simple idea with the how the nodes will be connected, the direction of the arrows, and the edge weights, which will be decided by the overlap length. Okay. Okay. So, as you can now see, right, we will utilize this the directed graph to actually get to the reference sequence. Okay. So, can we? Okay. Now, the idea is in the shortest common superstring approach is that we can determine the reference sequence, or we can derive the reference sequence from this overlap graph. Okay. Now, what do we have to do if we want to do that? What are we going to do? Okay. So, how can we find the shortest common superstring? So, the idea is if we can find a path that traverses through every node in this graph exactly once and maximizes the total score. Okay. So, you can actually traverse through these graphs. Okay. So, you can maybe let us say start from here, you can go in this direction, then go in this direction, and so on, right. You can see like there will be multiple such paths, right. So, you have so many options at every node, okay, and you can take any of those. What SCS approach tries is like find a path that traverses through every node exactly once and maximizes the total score. So, why every node exactly once? Because we have read, right. So, we are using this one read, like read only once, right, not multiple times. and the total score is calculated by the sum of the edge weights, right. So, if you are traversing through, let us say edge weight 4, then edge weight 5, you simply add these, right, 4 plus 5 plus and so on, okay, and you get the final score. Okay, so how do you actually find this path? Okay, so what we will do, we will transform this, right, this score into something else, right. So, we can think about something called cost instead of these edge weights, okay, or scores, and we can define cost as minus of overlap, right, just change the sign. Okay, we are not doing anything, just changing the sign. Okay, and the graph will look the same. What we have done is we have changed the sign of these edge weights, right. So, they are negative now. Okay, and what we have to do is we have transformed the problem, right. So, this means we have to find a path that traverses through every node exactly once, but also minimizes the total cost now. Okay, instead of maximizing the total score, now we need to minimize the total cost, right. So, we want as negative a number as possible. Okay, so that problem is transformed, and you will see why that is the case, because we see now this has a resemblance to something called traveling salesman problem or TSP problem. This is a very famous problem in computer science, right. So, this traveling salesman problem, and we know how difficult it is to solve this, etc. Okay, so, what is traveling salesman problem, right. So, you can think of this scenario, right. The name probably tells you that already, right. So, you can imagine an individual is traveling through multiple cities. Okay, so, again this is a graph of cities, right. So, city A to E, and these cities are connected by an edge, okay, because there are, let us say, flight connections or train connections between the cities. Okay. So, this individual, what he or she would have to do is to travel through each city exactly once, okay, and also minimize the cost of travel. Okay. So, you have to find an path, right, that will traverse through every node exactly once, but will minimize the cost of travel. So, the cost here are given by these W's, right, these weights on these edges, right. So, this cost would have to minimize, and the individual would have to travel every city exactly once. You can see this striking similarity with our problem, right. So, we want to traverse through every node exactly once, and we also want to minimize the cost. Okay, and this problem is known to be something called NP hard. Okay, so, without going into a lot of details what is NP complete or NP hard, it will be simple to say that this will take a long time as we start adding more nodes to the graph. Okay. So, for a small number of cities, right, or small number of reads, we can still solve it, right, within a reasonable amount of time, but as we start adding more nodes to that graph, this will increase very quickly. Okay. So, the time required to solve that problem will be quite long. Okay. So, you can imagine in our case, when you have millions or billions of reads, this is going to take a long time. We can think of this, we can simplify this, right. So, if we do not consider the ways, if we just say we just want to travel through every city exactly once, you can find these solutions, right. You can see like there are multiple ways to do this, and then it becomes something called an Hamiltonian path problem. Okay. So, again this is a famous problem in computer science, and it is known to be NP complete. So, again we are not going to go into the details of this NP hard or NP complete. What it means is that it is going to take a long time as we start adding more nodes to the graph. Okay. So, what we see is the shortest common superstring approach, right. So, it is going to be quite time consuming as we have more nodes to the graph. So, the question now we are left with is that how do we determine the SCS or shortest common superstring from the overlap graph in a reasonable amount of time. Okay. So, this is something we will talk about in the next class, and we will talk about other approaches in the following classes. So, here are the references for this class. So, to summarize, so we have talked about the de novo assembly problem, and I have discussed why this is a really challenging problem, what is the reason, etc. And we have discussed the SCS approach of the shortest common superstring approach. So, this starts with pairwise comparison of overlaps between reads. You will see other methods, one of the other methods that we will discuss also utilizes this overlap graph as the starting point. and then reads are represented in the directed graph with edge weights, which is again determined by the length of the overlap. The direction is also determined based on the overlap region, right. So, whether the overlap is in the suffix or in the prefix, that will determine the direction of the arrows. And finally, we have discussed, right, determining the shortest common superstring from the graph is equivalent to the travelling salesman problem and is an NP hard problem. Thank you very much.